The overall goal of this procedure is to isolate specific stages of mid to late Drosophila follicles for a variety of experiments, including molecular analyses, live imaging, and in vitro development assays. This is accomplished by first dissecting out the ovaries and then separating the ovaries into ovarioles. Then, once specific stage follicles have been isolated, they can be utilized for molecular analyses, live imaging, or in vitro development. Ultimately, results can show what aspects of late Drosophila oogenesis are regulated by specific factors through pharmacologic and genetic manipulations in combination with these assays.
This method can help answer key questions in the fields of development and cell biology, such as actin cytoskeletal dynamics, morphological processes, tubulogenesis, and extracellular matrix secretion and patterning.
For this protocol, collect well-nourished young adult flies of both sexes 2 days before isolating the follicles. If the staged follicles are going to be used for in vitro development assays, have freshly prepared IVAM media ready at room temperature. Cold media will alter cytoskeletal dynamics and development. If the staged follicles are going to be used for mRNA or protein isolation, Grace's insect media is sufficient. Now, begin by anesthetizing flies in their vial with an injection of carbon dioxide, and then transferring 3 to 5 females onto a carbon dioxide-emitting fly pad. Be aware that the flies cannot remain under CO2 any longer than 10 minutes without harm. Now,